Thank you very much. For us, it was really great to be able to participate and present this project. I'm trying to get my screen to play. So I will quickly switch. We're interested in brain anatomy, the origin of the shape of the brain. The shape of the brain is interesting and not just like some weird or beautiful artifact, we think. There's an increase in the complexity of the shape, in particular brain folding with brain size. That's pretty stable. This is probably one of the strongest correlations that you will ever see in the whole field of biology, right? Look at this slope, it's like a dream. And this geometry of the brain, the folds correspond to some degree with the architecture of the brain. Here you see different cytoarchitectonic regions, and it's not infrequent, even in a brain as folded as the human brain, that folds will have some relationship with the cytoarchitectonic organization. This is not only the case for the type of tissue, but also at the level of connectivity. There's structures that well described, like U fibers, for example, that would link one fold with the other. Okay. What's very interesting with the ferret, and that's why we proposed our project to study ferret brain development, is that the ferret, when it's born, it has a completely smooth brain. It's basically the size of a mouse brain. In a matter of 1 month, it will grow a brain that reaches the adult size, that has a pattern of folding that's very deep and very rich, similar to the pattern of a carnivore like a cat or some other carnivores. And it's also by that time, by 1 month, when they will first open their eyes. So they are like the last trimester of gestation in humans is equivalent to the first month after birth in ferrets, which is very interesting. So we'll continue. In particular, for human brains, the brain is already folded at birth, so it would be much more difficult to study brain folding in humans unless you are able to do MRI in vivo or something like that. So we had scanned previously a lot of ferrets, and with the funding that we obtained from HBP, we were able to do the histology for a series of brains, and then we put all this data in the FIIND website. The first page is what I showed you before. So we made available all our magnetic resonance imaging data from birth to adult, and also our histological data. That's a little bit fun, work that's still ongoing, because there's so much data that we need to digitize that takes quite some time. So if you go to this website, and I hope that I wrote the URL somewhere, you will have a table where you can select the developmental stage that you are interested in, and we have several individuals per time. And then there's also the possibility of do the same with histological data. And we also have, so the FIIND website has an API that you can directly query with Python code, for example. So we have some example notebooks that can show you how you can query data that's already present in BrainBox or in MicroDraw, which is our tool for MRI and for histology. And then you do your more fancy analysis offline. So this is the example of just one simple notebook that would let you download the data, but more interestingly, the annotations on top of the data. And that's what I would like to show you in a second. So using this data and the segmentations that we did with the tools that that I will show you in a second, we were able, for example, to do this reconstruction of the developmental trajectory for brain folding in a ferret going from P0 to adult, and we are still working on trying to analyze all this. We are also trying to combine our data analysis with simulations of brain folding. And this is a biomechanical model starting from the shape of a brain P0, and then running some growth models that will then generate a folding pattern. And we're trying to understand the differences and similarities between this folding pattern and what we observe in real animals. So I will now try to, I will stop sharing to try to share a web browser. I wanted to just very quickly show you how all this looks like. So this is the FIIND website. You go to neuroanatomy.github.io/FIIND, where you get a little presentation of the project. And then what I showed you before, you can access the histological, the MRI data and the histological data. And I will first go to the MRI data and just pick a brain that would be nice, like, I don't know, this one. The brain opens in our tool BrainBox. What's interesting with BrainBox is that it, and I hope it will work. I'm sure it will. Still loading. We'll probably go to another brain that may be less difficult to download. So you just go in and log in with a GitHub account. It's everything we need. BrainBox. So the landing page is like this. If you have the URL of any brain MRI data set that would be sitting on the web, you can just paste it here and you will get a viewer and the possibility of annotating on top of it. When you are logged in, you get a user page where you can create your projects. And this is the list of all my projects. I will show you, for example, the FIIND project. So a project is organized like this. You get a list of collaborators, and then you assign them rights if they have right to view, to edit, to delete, etc., etc. Then you add a list of MRIs. They have to be sitting on the web. Our data from the project funded by HBP, we uploaded all the data to Zenodo, so that it would be in a nice, safe, open place. And this is the list of files. And then when you go to the project, you get this table on the side where you can see all the brains. And then here you get this is a segmentation of the surface of the ferret brain. I will just make it less prominent. And then you have a series of very simple tools, for example, for drawing. No, and what's interesting is that it works in real time. So if two people are drawing in the same brain at the same time, you will see each other as if it were a Google Doc, right? And then there's, well, tools, of course, for erasing, and I will erase because I don't want to leave traces of my presentation. And you can, for example, do a 3D render of the segmentation that you have thus far. And this is one of our 3D models. Well, just the manual segmentation of a ferret brain. You can, so the comments table works also like a Google Doc. So if you have a large data set that you want to annotate, or if you want to annotate stuff with the ferret data that we made available, if two people are annotating, you will also see each other, so that it's very easy to collaborate and work together. And that was our aim. And this is so the tool that we use for, currently there's more than 13,000 MRI data sets indexed through BrainBox, which makes it possible for anyone from their home to work together on the data. And this is the same site, but for histology. And I will go quickly to, I don't know, I will show you the brain of a gorilla because it's fancy. And this is the same sort of thing, but this time is for histological data, and you have this deep zoom viewer. And in the case of the gorilla, in collaboration with some other people, we have been annotating the cerebellum. And here you see segmentation of the cerebellum. So the cerebellum is very, very complicated, and being able to work many people at the same time in the same data set and just online makes it possible to do these larger projects. So again, here, if I go to Project FIIND, this is where we have, so all these annotations are available to anyone that would connect to the website. And what we have been doing is segmenting the cerebrum, annotating all the different artifacts, so that when you run your analysis, you can discard them automatically or eventually learn to distinguish them. This is just a few of the brains that we have, and from these, using the tools that we made available with the notebooks, you can, for example, generate a 3D model that would be registered with your histological data, and then eventually also register with an MRI data. And just to finish, I would like to very, very briefly present what was the consortium for FIIND. that should be here. Yeah, so this was a collaboration between my lab at Institut Pasteur and then the group of Paul Kiesinga at the Radboud University in Nijmegen, the Mirsen lab at CEA, and also the MNI lab with Alan Evans. And well, thank you very much.